Hello, everyone. Welcome. We will go ahead and get started in just a moment. Thank you, everyone, for coming today. All right, excellent. Welcome, everyone. Thank you so much for being here. You have tuned in today to our annual Research Week presentation. Today's webinar is one out of 4 webinars that we are bringing to you this week that are focusing on the latest in science, clinical research, and future ALS treatments. Today's presentation will be given to us by Dr. Nick Olney of the Providence ALS Center and is going to focus on some of the latest updates in current clinical trials. All right, we would like to just take a moment to thank our sponsors for helping to make this event and the services and support that we offer at the chapter level to people living with ALS and their families possible. So thank you to Mitsubishi Tanabe Pharma America, Providence Brain and Spine Institute, Pharma, and New Motion. And we do have just a short word from one of our sponsors, Mitsubishi Tanabe Pharma. Just a moment while I get this pulled up.
Health is precious, and disease can be a deadly adversary. Some diseases present us with seemingly impossible challenges, but we don't believe in impossible. We believe there is always a way to help people struggling with rare and debilitating diseases. Yes, it takes intelligence and courage. And yes, there may be disappointments and failures along the way. But we'll never stop pursuing the impossible. Because we know that one scientific breakthrough can change everything.
Great. All right. So just a few housekeeping items before I introduce our speaker today. Today's presentation will last approximately 40 minutes, and then we'll have time for a Q&A portion at the end. All participants' video and microphones have been disabled to reduce interruption, but we do encourage you to ask questions for Dr. Olney during the presentation, which you can use the Q&A box that's provided at the bottom of your Zoom control panel to type in your questions. I will let you know that we received those, and then at the end of the presentation today, we'll go ahead and answer those live. And then also want to let everyone know that today's presentation will be recorded and made available on the chapter's YouTube page for later viewing, and will also be emailed out to you. Without further ado, I'm thrilled to welcome today's speaker, Dr. Nicholas Olney. Many of you might know Dr. Olney as the clinical director for the Providence ALS Center. He completed both his medical degree at the University of California in San Francisco and his neurology residency at University of California in Los Angeles. He eventually returned to UCSF to focus on neurodegenerative disease and research and came up to join the Providence ALS Center. Dr. Olney's passion and drive make him an outstanding research advocate and clinician, and we are just really excited to have you on today, Dr. Olney. Thank you for being here. I'm going to go ahead and stop sharing my screen and invite you to share yours, Dr. Olney, and start your presentation.
Great. Yeah, thanks so much for having me today. I think, is your screen still sharing? No. Great. So let me know if this looks okay here.
Perfect.
See this? All right.
Oh, we have it up. Thank you.
All right. So yeah, so I'll just get started. So yeah, still doing these virtual presentations. Did a couple last year. So yeah, thank you so much, Cassie, for the invite to talk here and for the nice introduction. I'll just tell a little bit more about my own personal story too. So I was born up here at OHSU, grew up in West Lynn, but second grade, moved down to the Bay Area in California. My dad was a clinician and did my high school there. Then I went to UCSD for undergrad, and I didn't exactly know what I wanted to do. So I was kind of debating on PhD or MD, and I wanted to do some kind of research. And that's unfortunately when my dad was diagnosed with ALS. But we, and then And then after that, I was lucky enough to get into medical school at UCSF so I could still see him. And I was still doing some part-time caregiving. Then I had to go back to do all the residency stuff. So I did a year of medicine in Oakland, 3 years at UCLA general neurology, then went back to UCSF to do a ALS focused fellowship in clinical care. And I was doing research in dementia and ALS at the same time. Let's see. And guessing my screen is not going forward, but I'll talk about it as I troubleshoot this, but I kept visiting the Northwest and always looked at it as a place to come back to. So this is Black Butte, where I visit very often throughout my life every year. This is a picture one of our family friends took that I put here. I'm going to try to stop sharing and reshare again. I don't know why my screen's not working right now. There we go. Can you see that, Cassie?
No, we're not seeing it.
Sorry. Let's see here. Sorry for the technical issues. I haven't had these before. I'm going to just reopen PowerPoint.
It wouldn't be a virtual presentation without at least one technological issue. It just happens.
I think I just need to share the screen first and then open the PowerPoint.
Yeah, sounds good. I think you're starting. Perfect. Yeah.
And then let me make sure I can get to presenter. Can you see it now?
Yeah.
And now
Perfect.
it's moving. All right. Sorry about that. Yeah. So then I end up interviewing up here with Dr. Goslin and a very interesting clinical trials. So it's been a great match ever since. So that was my dad. Actually, he had been diagnosed, and this is me as a caregiver back when I was younger. So for disclosures, I have presented my work in pseudobulbar affect to Avanair, who came out with Nuedexta. And then I am a PI for these clinical trials that I'm talking about today, Healy, Combout, and Alexis and Valor today. So if you're one of my patients, you probably have heard me talk about this. I say this often, but I'm just going to give a little clinical trials 101 here and explain the different phases we have in our country. So phase 1 trials here, as you can see, is really like first time in humans. So there's been a lot of preclinical basic science data, but the phase 1 is really first time in humans and typically is about 50 people, sometimes a little more, a little less. These are typically not coming to the Northwest. Phase 2 traditionally has been different dosages and looking at safety trials, and that's where we then pull the preclinical data for the phase 3. And phase 3, as you've heard about, is both when you're having to prove that it works by giving some people drug and some people not drug or placebo. And that's the phase 3 randomized control trial that is our standard to prove if a drug works. So we're tending to get phase 2 and 3 here in the Northwest. So just to give some examples of that, Alexis is one that's actually pretty close to filled nationwide, is a dose escalation phase 2. So basically they'd have a certain amount of patients go and they would go up slowly on the dosage, looking at safety and then also monitoring clinical data. They would be enrolling patients 2 years from the date of diagnosis. It's just 3 months of infusions every other week, and it's an anti-CD40 ligand antibody, so it's kind of like a focal immunosuppressant that had some good data in the basic science. And this is all pulled from clinicaltrials.gov. So this trial is actually pretty close to filled, I understand, but I think this is an exciting study that I hope will go into phase 3. And another phase 3 study we currently have. So this is it's phase 2/3. I think it's a little designed a bit more like a phase 3. Just in that it's 12 months, 50-50, whether you get drug or placebo. But this was a compound that's quite safe. It's been on the market in some version in Japan for 20 years, called Abudilast or MN166, and lots of potential mechanisms it could work with a MIF inhibitor, phosphodiesterase inhibitor, even some basic science data of helping autophagy or eliminating bad proteins. And actually, there's a big talk that Dr. Bjorn Oscarsson gives at the Medica Nova site if you want more interest in this trial. I think they did have an early trial showing that some people in their earlier trials did not show progression. So they designed this trial with some very strict criteria to try to answer the question. So I think, although this is a challenging study to recruit for, I think it is good to have the actual answer to this question. So So this is just more of an example of probably a classic phase 3. Now, going back to our design of the phase 1, 2, 3, I mean, people are getting creative now, putting placebo into the phase 2 trials. And as you heard about the Centaur, and I believe you hear about that later this week, I mean, they're trying to get early FDA approval since that actually showed success with that trial. So people are getting creative, and I'll explain that here. And one of the most, probably most novel approaches has been the Healy platform trial, which we are a part of. So to kind of go into that, the traditional clinical trials, we build a big stadium, we run a race. This takes about 2 years, and then we kind of break the stadium down and get the results from the study. The platform trial here is going to make a giant stadium where we're going to run multiple races at the same time. And then as one race closes, we can open another race or therapy or regimen. So I kind of use those words interchangeably. And so this makes it a more efficient way to test multiple compounds at the same time. So to give a little bit more of a picture view of this again, first people, when they come to screen for a trial, they're going to come to just see, can they kind of enter the stadium? Then if they do, they pass all the safety criteria, they then are assigned a certain regimen. And then they're then assigned and screened for that specific regimen and assigned either active or placebo. And so one of the exciting things, or at least what patients I'm also excited about, is it's a 3 out of 4 chance of getting active drug and 1 in 4 a placebo. So that's enriched a lot more than compared to the traditional trials. And that's also because they're able to share placebo across the different arms of the study. And then the other thing that I think is also enticing for patients is that it actually goes open label at 6 months. So people like that they have more of a chance of actually getting on one of these active compounds that are in trials. And so they've also done some really powerful statistics. I sat through a lecture this at the international conference in Perth, and some pretty complicated statistics, but it looks pretty sound from what I could understand. But they are able to cut the costs in a third, cuts the time in half. Again, I guess it's kind of similar. I've already explained it with the other picture, but the less placebo has a lot of complicated statistics behind it, and that's why you have to enter this trial. You have to really be okay with the randomization part of it. That's why I'm taking the time to explain it here. So we're also looking, the statistics are planned to detect a change that's greater than 30%. We certainly want something even better than that, but this is designed to check something that's a 30% change, which would kind of be the thing to beat these days with Radicava on the market. And so they also have done some just statistical analysis that if there's 10 therapies tested, we can cut down the amount of participants by dramatic time and the amount of time and the amount of people on placebo to get answers about these compounds and to get results of something if it shows a 30% change. So I think that's quite exciting. So here are the compounds. As you see, there actually are quite a lot of different mechanisms and a lot of different pharmaceutical companies. So, I mean, you could talk a lot about each of these. I'm just going to probably talk about them briefly, but they're also very different kind of mechanisms. So the zilucoplan, it's a complement inhibitor. So kind of, and it's an injection. Rosuvastatin, myeloperoxidase inhibitor. So that actually turns off part of the cell. We think will do good things for neurodegeneration. The CNM-AU8 is a liquid and it's a gold nanocrystal. So it's involved in kind of a major antioxidant, kind of involved in the NAD pathway. Pridopidine is a sigma-1 agonist. So again, kind of turning something on, we think will be good for neurodegenerating cells with eliminating proteins, providing protection. And the CD14, this is actually an infusion medication, kind of also targeting immune pathways. So you can see there's a lot of variable mechanisms. So I think some of the challenge with the Healy is that you have to be okay with the kind of randomness. But all these compounds are chosen by Harvard. They all have promise, and they all have also have biomarkers that we're also planning. So besides just the clinical data, monitoring those as well. So we do measure some of the standard endpoints of the ALS functional rating scale, which is pretty standard across our trials. And they're also measuring breathing changes, the handheld dynamometry or the grip strength, survival. And then they also, as I said, all have specific biomarkers for each compound to look for target engagement, which I think is also a really exciting thing to be having added on to a trial. And there's some exploratory endpoints. And then of course, we're always looking for safety. I mean, all these trials are really designed for safety. And then, so here's some more of the endpoints we have. So there is possibility to bank the DNA at the Harvard Biorepository. I would also just point out they have some really interesting things with speech digital. They're already planning that pre-pandemic, but that's really been helpful as where I think clinical trials might go with kind of checking for voice and a whole lot of home spirometry and things like that. So quite exciting. So the Healy platform trial, as I mentioned, the first 3 are pretty much filled. So pretty soon we're only going to have these last 2 open for enrollment, but this has been a very exciting trial to be a part of. It's actually filled about 3 times faster than any prior ALS trial. And it also, I mean, I think with that it filled so fast, we're going to have results quicker. I mean, some of these might, you know, if the regimen's filled, I mean, probably about a year or so, we probably have results, which I think is really exciting for the ALS community as a whole. So moving on, I think one of the other most other exciting things in clinical trials is ALS genetic therapies. So I'll come back to this picture in just a second, but I'll give a brief primer on ALS genetics. So think about ALS genetics, about 10% is being hereditary. Like these are kind of some of the main players that we're finding more and more genes every month, every year. But probably the biggest ones that are actually have compounds with some intervention would be the SOD1, which is overall 2% of ALS, and then the C9orf72. And I think that one also has a prevalence rate is up to into sporadic ALS, so it might not have a strong family history, and people are finding this gene and might have a potential treatment. So I think that's why these are exciting things to hear about for ALS genetic therapies. So just to tell you a little bit more about the story of SOD1, this is, unfortunately, people have SOD1 mutations. It's thought to be a toxic gain of function, toxic to have some of these mutations. Basically, it's thought that the DNA is creating a toxic protein that causes ALS. And often if you have this and people live long enough, they do express ALS. And often it's about 50% of family members have this. It's a very devastating mutation to have. Now, the idea, though, is that if we know a target, that maybe there's a way we could silence this target. So to just go kind of through one of the dogmas of molecular biology, we have the DNA, and this translates RNA, and then the RNA then translates the protein. So there's a way that we could put in an antisense oligonucleotide, which is a mirror image of this RNA, and block it at the RNA level here so that the bad protein is never made, and hopefully this will slow down disease. So I've been hearing about this for about 20 years. This is Tim Miller, and I actually shadowed him when I was at UCSD deciding kind of what I was going to do. And he was learning from Don Cleveland at that time about this technology at the basic science labs. And he's one of the main clinicians that gives a lot of our talks. So that's why I showed his picture. And I also have a personal connection as well. But there's a lot of people behind this technology, but it's taken about 20 years or so to get it into a safe way for humans to be able to get this drug administered, to try to minimize side effects and to try to prove if this works. So of course, there's a lot of more people involved. Tim Miller is obviously one of the big clinicians giving talks on this, but Don Cleveland, Toby Ferguson at Biogen is also very involved in the biotech side of pushing this forward. I mean, there's a large team of people, but I also mentioned Tim Miller because he gave a recent very exciting talk about the phase 3 results of Valor, which I'll talk about here in just a second. So it's both, they recruited both slow and rapid progressors, and we're actually giving the SOD1 ASO, and it's a trial that we've had open here at Providence for the last year or so. So this was a really just hot off the press, just presented at the American Neurology Association on October 17th, so just 3 Sundays ago. I'm just going to cherry pick some of the highlights. Now, I did contribute data to what actually comes out in the group data, but I did not do the actual crunching of the data, and I did get permission from Biogen to share these slides. So, and you can actually watch the whole presentation yourself if you wanted to know more. It's at the Biogen website here, but I fully give them credit for designing these slides, but I got permission to present them. They also do use some language that these is very future thinking and speculative, but I think the data is quite impressive. So, so to just kind of go a little further into this slide, and this was presented by Tim Miller, October 17th again. So the study was enriched for fast progressors. They recruited patients with SOD1 mutations that had 1 point or greater decline per month since symptom onset, or patients with these A4V mutations, which typically have a very fast progression and prognosis of 1.2 years from symptom onset. So a very fast progression. And then they also, so 60 patients with this fast progressing group, and then another set of patients here, 48, with a slow progression. A little less stringent criteria for the slower progression. And then they're randomized 2-to-1 for getting the tofersen and the placebo. And now this also is given intrathecally, which is actually a lumbar puncture that then you do a 2-minute push of the drug over 2 minutes. So, so you do have to be okay with the lumbar puncture part of the administration and for this to get this drug. And what they measured was the ALS functional rating scale. Then they also measured the breathing of the slow vital capacity. Again, the grip strength, time to death. They also had some really exciting fluid biomarkers of the SOD1 total protein and neurofilament, which I'll talk about in just a second. And again, I kind of just cherry picked about probably half the slides to present, just mostly I think for what patients would want to know and family members with ALS. I didn't go into the whole presentation, so I would recommend going to the Biogen site if you want to see the whole thing. But basically, the characteristics, so I'm not going to go into this in a lot of details, but basically it was well balanced, but between who had Relutek, who had Endaravirone, pre-slope before entering the trial. And so the ALS functional rating scale at the 6-month time point for the Valor, unfortunately, did not show significance. So here's where you can see this data of just the Valor data. And so just to walk through this, the red line is the fast group, and a solid line got the drug. The dotted line is placebo. Blue is the slow progressors, and the solid line got the drug, and the dotted line is placebo. So here the graph, we can see the slow progressors here, and we can see the fast progressors. And at 6 months, it's hard to say that the lines here, this shows the, the standard error, and you could not statistically significant. However, I think it starts to get interesting when you start looking at the open label portion. So when you expand this into the open label, I think especially when you start looking at the slow progressors, you really start seeing this possible stabilization. The natural history of this initial group, they could have always just been slow progressors. But when you see a slow progressor starting to decline, then they start getting drug and it starts looking more flat. I think that looks pretty exciting, even if it's not statistically significant. Now, to go for the target engagement. So again, to prove that we're engaging the SOD1 protein, this is just a general, again, same solid lines getting drug, dotted lines placebo, and again, red being fast and blue being slow. Again, we see that here the placebo groups with SOD1 protein staying pretty stable, and the SOD1 protein in people getting the drug going down. So I think that's just showing good target engagement. And we see this both in the Valor 6-month study as well as the open label. And if anything, you might even see more of the protein going down here as it gets in longer, or those who started earlier. Then the effect on neurofilament, again, this is thought to be, neurofilament is probably more of just a thought of neurodegeneration. It's not specific for ALS. It can happen in other dementias and other diseases, but it is thought to be a sign of basically neuronal death. So this is actually, again, the Valor showing actually a pretty significant decline in neurofilament. So I think it's actually quite impressive. I'm not aware of any other study that's shown this much decline in neurofilament. And again, we look at the Valor for 6 months and then the Valor plus the open label, and we again see this similar trend of the neurofilament going down and staying down throughout the time of getting the drug. And now when we look at the clinical measures, so this is the breathing, I think we again see the trend here at the slow progressors of people that were trending down, and then after they start getting drug to have this potential stabilization, to even at least declining the rate of progression. And then same when we look at the grip strength of the handheld dynamometry, not easy to separate here, but as we look at the open label, I think we see this trend again. And then the other thing that I would say also for the rapid progressors is that I mentioned there's this was enriched for fast progressors and patients with these A4V carriers, which typically has a unfortunately life expectancy of 1.2 years from the date of onset. And in looking at the natural history, there's only been 2 reports of people living over 3 years. And here from the pulling the group of the A4V carriers, there's actually 4 that are still living, and 1 is actually at 3.2 years. So I think it's also showing some exciting potential target engagement and effects here. So when looking at the safety, I think overall was quite safe, and a lot of the side effects more due to the multiple lumbar punctures, headaches, back pain. Probably the main neurologic thing that happened in, again, 1 of 100 patients was a mild to moderate myelitis, which was, I heard, managed clinically, and there's 1 report of meningitis. We also started noticing some changes in the CSF with increased protein and leukocyte shift, but we thought that these were not symptomatic. So I think overall, but overall the side effect profile quite good, especially given its lumbar punctures. So in summary, again, from this presentation, we do see this effects become more apparent of the tofersen with long-term extension, and when it started earlier of slowing the change of the fast progressors and the slow. The tofersen showed good target engagement with the SOD1 protein going down and the neurofilament going down, and the side effects were mild, with the 1 serious neurologic side effect being the myelitis, which is thought to be mild to moderate. So again, this is actually a really fresh set of data. People are still looking at it actively, and this is really just top-line data that just came out a couple weeks ago. So very excited that that Biogen let me at least said I could use some of these slides. And then they actually had a thank you slide, and it's very exciting for me, again, as someone who's had a family member with ALS, to be a part of this exciting study and just be mentioned as a thank you here in the thank you slide at this big conference. So. And then one other thing, I think if this is really exciting for SOD1, although this is a small portion of overall ALS 2%, there is a drug already. I don't think it has a fancy name that I'm aware of, but at Biogen for the C9orf72, that might be applicable then to those sporadic cases that would have a gene mutation. So I think that's actually very exciting, because if one of the genetic therapies is successful, then even more, I think, would be coming down the pipeline. I always look at clinicaltrials.gov, and I always see this number increase. So see how much interest there is in ALS clinical trials. If you just search ALS on clinicaltrials.gov, you find 895 studies with 166 actively in recruiting. So I think that's very exciting. I pretty much just told you about most of the ones at the Providence, but we haven't had more studies that I was not able to mention today. And with that, I just want to thank you for your attention. Thank you for the invitation to talk. And again, I always say this, but I'm hoping someday that we can make the ALS centers ALS treatment centers. So thank you for your attention.
Great. Thank you so much, Dr. Olney, for your great talk. That was really, really informative, and we've had some good questions come in. Would also encourage folks who have not to ask a question, if you're interested in submitting one, you can do so in the Q&A function. But because you were just talking about the results from the Valor study, I'm wondering, of course, we've heard about Valor and some other trials recently that have had, somewhat disappointing results or have not met their primary endpoints. And I'm just wondering if you can talk a little bit about what that might mean for potential therapies. Is this all of this information that we've gained from these trials kind of moot at this point, or do you think there are other avenues where we can gain further information from the results of some of these studies?
Yeah, so yes, I guess I'll start with that. I mean, I think that every study we get, when they're well designed, we're getting an answer that we at least have checked something off the list that we know it does not work. I mean, I think the other thing too is, I think I always talk about this with my patients considering trials, is that, you want to make sure you have the bandwidth for it. These are all experiments. We've actually had 3 positive trials, which would be the Riliatech, Radicava, and the Amylyx. And I think then we're starting to look at these other secondary endpoints. So again, actually to point out the Valor, I was there with a group of all the PIs, and even though the ALS functional rating scale was not significantly changed, we actually are wondering, well, should we be going off of these secondary endpoints as well. I mean, if you look at the ALS functional rating scale, it is a validated scale, but it it captures different regions of bulbar or speech swallow changes, arm, leg, and breathing changes, but it captures a very heterogeneous group. I mean, you can see these spaghetti plots, and it's very different. So it's a very challenging disease group to study. I mean, I think if we're able to stabilize the ALSFRS, then you're very confident that your drug is working, but there's these other things that also hint that they might be working as well. So I guess I would say that, I mean, I think you always have to set up realistic expectations, but I still think there's a lot of hope in these trials. I think they're getting, I mean, as the reason I show that slide of clinicaltrials.gov, there's a lot more people. I mean, even back 50 years ago when my dad had ALS, I mean, there's so much more interest in ALS, and both in academics and in biotech right now. I think is an exciting time to be entering these trials. So I think just unfortunately, we do have the failures. I mean, we obviously want the treatment, we want the cure, but I think every unsuccessful trial, we have an answer that gets us closer to the next one. So I think that's what's exciting. And I think you do have to use those phases, though, for the risk assessment of the trial you're signing on for. So that's why, again, the farther you're on into phase 3, probably the safer it is. They're never totally safe, but the phase 1s tend to be higher risk but you're potentially getting the first drug, ever in the human kind of. So answer your question.
Yeah, I think so. There is kind of a similar vein. What specifically to the Valor trial, from the data that you've seen, what do the results sort of suggest for people with sporadic ALS? Is there any information in the data that looks promising for the future for folks who don't have the SOD1 mutation, whether it's another familial gene or just sporadic?
Yeah. So I would say that I think we just see that from the target engagement data we have. I mean, the genes look like we have a target, and I think we have a safe way to get there to the target. I think the thing I would think about is probably it wouldn't be all the sporadics, but the C9 or 72 is something we do see in patients that don't have any family history, and we're finding this kind of more spontaneous mutation, and that would have a potential treatment. So I mean, I guess some of that's reported up to maybe 20%, and sometimes even higher in sporadic cases, but that would be a potential treatment, and I think that's one of the reasons for having genetic testing actually be more common in practice. I mean, obviously it's loaded. You want to have people with genetic counseling, but I mean, I think that would be, I mean, where do we have the best target engagement are these genetic therapies is what I'm seeing. So, and the sporadic, I mean, I think these other trials are all really great mechanisms. I think we're getting smarter. We're having much more mouse models and different things that we can look at now. I think that those, though, are probably more broader spectrum that we think would apply to probably both genetic and sporadic. So, but that's what I kind of trying to put in my last slide there, that, I mean, I think if the SOD one, although that's only 2%, if that's successful, I think they already have a drug in the pipeline for the C9 that would probably potentially spread to a certain percentage of sporadic. But I think the thing is we need to really increase the amount of genetic testing we're doing in the majority of sites. I mean, I think there is going to be a Biogen sponsored genetic testing program coming out. It's already available in some places. We're probably going to have this here pretty soon at Providence. But they actually know that that's an issue in the world of ALS. And I would also say compared to the world of dementia, it's probably much more common to have the genetic testing done. So I think that's kind of the avenue we're going.
Great, thank you.
Question.
Yeah, thank you. So there were a couple of questions about clinical trial access. So you kind of talked about this a little bit, but what recommendations would you have for individuals to get involved with clinical trials on either a local level or say they're living down in Southern Oregon or way up in Battleground? How might they get involved? And then kind of a part of that, what would you recommend for folks who maybe have a PLS diagnosis or a PMA diagnosis or just aren't qualifying for a lot of the eligible clinical trials?
Sure. Yeah. So to get involved in the trials, I mean, I think we're one of the biggest clinical trials in the Northwest, but there are other sites like OHSU and UW and Swedish here in the Northwest that have trials. I do think when you're traveling a distance that is something you need to take into account and that's part of the bandwidth I kind of talked about. So, I mean, generally these are designed for safety. So you're generally needing to check in at least every 4 to 6 weeks for some kind of labs or monitoring. I think with the pandemic, we've actually gotten a bit smarter about how we could do more home monitoring and home safety. I don't think everywhere is quite to the point though that they would have traveling nurses going to more remote locations. I mean, I might get there someday. But for those that are interested, I mean, you can always reach out to my clinical trials team, Ashley Adamo, and we have a big trials group. And again, any of those other places that are doing trials, they're all available as well to recruit people. Now, I will say that you're also right, Cassie, that usually they're wanting very strict criteria. So my job as a PI is to make sure that the company or the company or sponsor gets the quality high-level data that they want. But also my job is to honor the patient and safety. I mean, I really have to make sure someone's getting the medical care and that these experimental compounds aren't causing any serious adverse events. So with that, I mean, you do need to count on the safety. Now, for those patients that don't qualify, so again, going back to the main criteria, it's generally based on breathing from the date of symptom onset of weakness. So twitching and other things don't really count, but weakness. And usually the most they've ever included is up to 36 months. And that's actually been the Healy and the TAME trial. Most of them have actually always been at 24 months. And actually the Analexis is one of the first ones that I've seen that actually has included from date of diagnosis. And so the symptoms could be longer. So is that the right way? I mean, there are people in a lot of the conferences debating that, but that's kind of how it's set up right now with the rules. I mean, my job is I just have to obey the rules set out by the sponsor. I think when people are starting to not qualify, I do think that some of the national meetings, we've talking a lot about more open access or expanded access. I'm not aware of any of those coming to the Northwest right now, but those are certainly, they're starting to pop up and certainly something all of us are thinking about. I would also say one of the big people that I always look to and try to talk to when I'm at conferences is Dr. Bedlack out of Duke. He is a very all-inclusive, and I really try to model my clinic off of that too. And he writes the ALSuntangled.com. So I think, I know I've talked a lot about with patients for Amylyx or also some of the ALSuntangled.com kind of compounds. I always think also talking about the FDA approved treatments. I mean, I think that even when people are PLS or PMA, you have to tell them that the data is pretty limited for those groups. I mean, unfortunately, that's just a rare enough diagnosis. It's hard to always know how all these things apply to them. And that's why it's challenging to get in. I think you just have to have a long in-depth conversation about these things with patients and tell them kind of what they're at. I mean, I often say that too. I mean, sometimes when people have more of a PLS, PMA diagnosis, that's generally already thought to be a slower progression, but it also means they can't get into trials. So I tell them pretty upfront about that. And I certainly, if there's any compound I hear about that I think would be available and you could give it to anybody, I try to let people know as soon as possible. And actually so one other thing to add in, I mean, with those Valor results, I certainly told the patients we have involved in that as soon as I could tell them about it. So you want to be as open as you can. So.
Great, thank you. I had a similar kind of question, which you've already sort of answered about alternative and off-label treatments. And I did put the ALS Untangled website in the chat for anyone watching. I just curious if you want to speak a little bit more to just your feeling about off-label or alternative treatment options. I know there's so much out there to look into and investigate. What would you advise patients of when they're sort of muddling through all of the options?
Yeah, I mean, I think that I always think this is a conversation. There's not one size fits all, but I think certainly I always try to make sure someone's educated about the FDA approved treatments, which would be the Realitech and the Radicava. Then I kind of approach about trials and experimental compounds. And I think the ALS Untangled does a really great job of presenting safety data. I mean, again, I really would hate for someone to get a compound or one that's not safe, and two, spend a ton of money on something that we just don't have any evidence that works. So those are the first things I usually look at. Dr. Bedlack actually started to be involved in so many of these trials. I would say that anything that really has a lot of great kind of reviews on ALS Untangled, usually you can't be on that and be in a clinical trial. So there is sometimes a conflict there that you have to kind of decide, or that is sometimes when someone doesn't qualify, that's when we kind of start to talk about. But like, for instance, curcumin is something that he's planning to, Dr. Bedlack's planning a trial on in the next year or so. A lot of people are doing it off label. It has a very good review on ALS Untangled and it's cheap. And so I always say that's one that I say, hey, worth a try. I think the challenge with a lot of these ones, except for people like Dr. Bedlack, there's a lot more money obviously put in marketing than the actual research. So Dr. Bedlack's one of the main ones I know that's really trying to get these crowdfunded even sponsors. Well, he'll actually raise locally at Duke about $200,000 and do a trial of 50 people to see if there's a signal there that it works and that it's safe and stuff. So I'm certainly open to all those things. I think it's I think there is a little bit, and sorry, one last thing on this. So I do think there's a little more conflict now with all these alternative or supplements kind of gaining steam, that people are excluded now. Like if you're on TUDCA, if you're on theracurcumine, you can't get into most trials. So that's some of the challenges.
Thank you.
Question.
Yeah. One more question for you. Well, actually, two more. You mentioned biomarkers a couple of times during the presentation. I'm wondering if you can just talk more about why biomarkers are important to ALS research, why they're included in clinical trials.
Sure. So biomarkers, I guess that, they serve as a way of probably, measuring clinical progression or stabilization. I mean, I guess even the exam or things like that serve as that. I mean, we could always look at strength and breathing as that, but we're usually, if we want to go even deeper, I mean, for dementia, we're often using imaging for, I think ALS, for instance, one of the blood samples, the neurofilament is gaining a lot of traction. And I think that's one too, that, you asked what would apply to some of these other more sporadic disease trials. I think that would be something that certainly is a hot biomarker, because I think it's getting pretty widely accepted. There's a lot more data that we think of it as kind of almost a neuronal death or inflammation going on. And if you lower that, it's probably a good thing. So I think that might start being even a secondary endpoint. So if someone has a drug that's able to actively lower the neuroinflammation, that might be a way of proving that we think it's doing something more at this molecular level of stabilizing neurons. So, but you always have to look at the strength of those biomarkers. I mean, what I mentioned for the Healy trial is they're trying to choose biomarkers that prove target engagement for that specific compound. So a lot of these are still targeting very broad mechanisms. I mean, sometimes they're the actual target in the cell, sometimes they're just very broad as inflammation, sometimes they're a bit more like free radical. So there's a lot of different ways to try to measure those, and it's just trying to get more data beyond than what we're kind of measuring from just the clinical exam.
Great, thank you. I think that was a really great response to a very big, broad question.
Yeah, there's a lot you can do with biomarkers. I mean, I always thought I was doing imaging researcher, and I said I want to do biomarkers. I was really excited I was trying to do one of the first neurofilament kind of studies in ALS at UCSF that I was aware of, and the imaging was actually a little easier to try to get going, just because I was more in place at the time when I was there. So, but I think I was trying to do a little bit of both. But I think that, Dr. Benatar out of University of Miami, who we've joined his research efforts, I mean, he's really been one of the big people pushing that biomarker forward. So.
Great. Well, you gave us a really, really great rundown of, kind of the latest in clinical trials and treatment options and kind of where things are moving forward. I always like to just end with kind of a hopeful question and ask what you're most excited about, or how you feel like things are kind of moving forward in the ALS research space in general.
Yeah. Well, I tried to choose all the ones that I'm really excited about to talk about today. I mean, I think they're all they're all different. And I mean, I think although the Valor, I think I'm pretty excited about those secondary endpoints. I mean, I think we really want to see where those go. And again, I think that's something I'm quite excited about, but all those are really exciting. I mean, I think the Healy is very excited to be a part of. I think the more, some of these really newer compounds in phase 2, phase 3 are also exciting. I mean, I think also I couldn't do this research if I didn't have patients that are willing to trust us to do these projects, and we wouldn't have answers if people weren't willing to do them. So, but I think that again, I mean, I guess bringing back to your other question is, we've had a lot of failed trials. Unfortunately, we've had more failed trials than successful trials, but I think every failed trial we have, we get closer to actually trying to figure out the actual answer for what will work. And I guess you also, just to tie back out of your questions, I mean, for patients that can't qualify for a trial, I mean, one of the designs of the Healy trial, we get an answer so fast about a drug in a year, I mean, that will spread to the whole community of ALS. So I think you have to have some kind of altruistic feelings you get from participating in these trials to want to participate. I mean, you obviously hope that you get the magic ticket or the magic racehorse for the drug you're getting, but you have to have also belief that you're helping the community as a whole. And that's one of the reasons, that's one of the things that keeps me going. But it's hard for me to always just choose one, because I have to kind of hope in all of them, because I recruit for all of them. But I mean, I guess I also say, I think definitely as genetic testing gets more common, and if you that would be probably one that I'm really excited about for that. It is a bit more invasive to get those drugs though. I mean, so far that's not oral, it's all lumbar punctures. So that also takes a certain person that's willing to do that. So.
Yeah, it's really a community-based effort, isn't it? It takes everybody's involvement from, the clinics that are hosting the clinical trials to the patients that are enrolling to the scientists who are working in the lab. It's just, that's really.
And I always, I guess that's some of the challenges. I have to be kind of a fan of all the trials, but I'm always trying to have different ones that might meet all the interests of all the patients I have in my clinic. And so like if Dr. Bedlack had one that expanded here, I would definitely love to be a part of that. And that would be my ones who maybe for the patients who don't qualify for that traditional from 3 years from symptom onset kind of trial. So, and then I'm always trying to be a part of other consortiums or genetic research or other things that would also just be part of research for my patients that want to be doing research too. So.
Well, we really, really appreciate your time, Dr. Olney, and coming and speaking with all of us today and answering all of the questions that we have. Certainly, if folks have other questions, feel free to reach out to myself and we can help get those answered. Actually, beautiful timing in terms of talking about just the growing interest in ALS research. For those who are able to log in tomorrow, we are going to be joined by 2 postdoctoral fellows who are actually funded through the Milton Safenowitz postdoc fellowship program through our chapter. And they're actually going to be both presenting on some really interesting topics. Dr. Nishal Shah will be talking about intracortical brain-computer communication interfaces, which for those who tuned in yesterday, we talked about some brain-computer interface work that's being done at OHSU. So that's going to kind of continue that conversation. And then we'll also hear from Dr. Lauren Giddings, who is from the Barrow Neurological Institute, and she'll be talking about cells in the brain that contribute to ALS and FTD. So I think there's going to be a lot of really good information for the rest of the week. And Dr. Olney, just want to thank you again for coming today, and thank you to everybody for tuning in. And today's talk will be available on our YouTube page later on after it's uploaded.
Thanks so much for the invitation to talk.
Yeah, thank you very much. Everybody have a wonderful
exciting week.